Okay, so what we're looking at here is membranes in action. What you may notice by the picture on the front of this PowerPoint, and that's one thing with every PowerPoint that you give you guys, always have a look at the front picture and see what information can you automatically get. So what we've got our phosphate heads, okay, little lollipop tops, the lipid tails that attach to those. Remember, 2 lipid tails for every phosphate head. We've got carbohydrate groups. So glycoproteins. Glyco, similar to glucose, so a sugar of some description, and the protein is just a protein. We've got our proteins that are embedded in the plasma membrane, as well as these protein channels. The other thing you might notice, on the top, we've got the extracellular fluid, so that means on the bottom is the intracellular fluid. So whenever you look at a plasma membrane, have a think about what information can I get before actually reading any text that may accompany it. Okay, so hopefully this kind of stuff is revision from year 11. So what you should already know about a membrane: they define the cell boundary and they provide a permeability barrier. So if something is permeable, it lets some things in, but not all things. It has sites for specific functions. How do we know what the specific function is going to be? Often in biology, things are very shape specific. It regulates the transport of solutes. It detects electrical and chemical signals, and it assists in cell-to-cell communication. That will be particularly important when we do the immune system later on. Okay, so this slide hasn't transferred over very well from PowerPoint, but it is exactly the same picture as what is on the first slide, so you should have some idea of what it looks like. Okay, so when we think about a plasma membrane, it's what we refer to as a fluid mosaic model. So fluid, it has some movement. Mosaic, it's not uniform. So lipids are important components of plasma membranes. They don't set the plasma membrane hard. So if you think of something like vegetable oil or olive oil, it's a fluid type of oil. Your plasma membrane is the same. So the lipids provide a fluidity to the membrane structure. The proteins are the mosaic part of the membrane, so they are interdispersed or placed in random locations. The proteins have a variety of different jobs. For some, they'll be assisting in facilitated transport, some will provide protein channels, some will be glycoproteins. So when we think about proteins and membrane structure, it's not a one-size-fits-all type of thing. Here we have an example of the different types of proteins that can be part of a plasma membrane. So it makes sense that the hydrophilic portions, so the water-loving portions of a protein, are exposed to the water on either side of the membrane. So the outside of the cell, the extracellular fluid, the inside of the cell, the intracellular fluid. Some of them can extend all the way through the phospholipid bilayer, and they would be a protein channel. So they will open and shut as the cell needs a larger molecule to come in, such as a protein or a glucose molecule. Some are embedded partway into the bilayer. They may be part of cell recognition. You've got some that act as facilitated transport, so a protein receives a signal from the environment outside of the cell, and then it will allow them to pass into the inside of the cell. Some proteins can move laterally through the phospholipid bilayer, and some form channels for ions and molecules can enter and leave the cell. Okay, so when we talk about selective permeability, and this is where I liken a plasma membrane to a fly-by screen. Some things come in, some things can't come in. With your plasma membrane, the 3 things that it is selectively permeable to that you need to know this year are size, degree of polarity, and charged atoms, otherwise known as ions. So the plasma membrane is permeable to small molecules. So oxygen, carbon dioxide, ethanol, and water are small enough to sneak through the plasma membrane without requiring energy or assistance. They are permeable to hydrophobic molecules. So if something is lipid soluble, it can pass through the plasma membrane unassisted. It is not permeable to larger hydrophilic, so big water-loving polar molecules, for example glucose, and charged groups of atoms and metallic ions. So when you do your SAC, it's important that you think about what solutes we are using and whether or not they will be able to pass through the plasma membrane assisted. So if they are not small, if they are hydrophilic, or if they are large, chances are they won't be able to pass through the membrane unassisted. So to move something through the plasma membrane in either direction, so either coming into the cell or leaving the cell, there's diffusion, which is passive, no energy required, or active transport, non-passive, so requires a form of energy. When we talk about cells, energy is in the form of ATP or adenosine triphosphate. Okay, so simple diffusion. You need to know a textbook definition for this. This type of question does turn up in exams. So it's the net movement down the concentration gradient to achieve equilibrium. From about year 7 science, you would have learnt that particles are always in motion. So the random collision of these particles enables higher concentration to move to lower concentration without any input of energy. So if you look at the pictures down the bottom, on the left-hand side, I've got very concentrated particles over here. Let's say, for instance, that's oxygen. Some time passes, and after a while, I've got quite a uniform distribution. Once I've reached equilibrium or fairly uniform distribution, you must remember that particles are still moving in either direction. They don't just stop. Particles are always moving, and that is one thing that kids get tripped up on in SACs and in exams. Okay, so with facilitated diffusion, if you facilitate something, you help it. So facilitated diffusion involving a plasma membrane means that it needs some help. So it can occur via protein channels or carrier proteins. So the top picture, I've literally got almost like a tunnel, and my larger molecules, so let's say glucose, can pass through this tunnel. It's made of a protein and it's embedded within the phospholipid bilayer. On the bottom picture, I've got what we call a carrier protein. Now, I sort of think of these guys like Pac-Man, because to me that's what they look like, probably showing my age there. But, so the top side opens, solute molecule pops in, top side shuts, bottom side opens and releases the molecule. The difference between a protein channel and a carrier protein, there is much greater competition for available sites. So I can only take through one molecule at a time. Once it's released down the bottom, the top then has to open up again. So that can actually slow down the process of diffusion depending on how many protein channels or carrier proteins are available to bring in the solute molecules. With osmosis, so osmosis is a form of diffusion, but in this instance, it is the solvent. For Year 12 biology, the solvent will always be water. The water is moving from a region of lower solute concentration to a region of higher solute concentration. So I'm going from a region of low solute concentration, so not much cordial, to an area of high solute concentration, so lots of cordial. So if you think about it, if I want to dilute cordial, I add in water. Okay, and same with osmosis, I've got too much cordial, so I add in more water to dilute it. The picture that you've got here, the thing to think about, so I've got net movement of water going towards the right. So it would make sense that the water level on the right-hand side would increase, whereas the water level on the left-hand side would decrease. So they're always things to think about, and I think that's come up in one of your chapter questions recently. Osmosis occurs when the particles that make up the solute, so think of our biomacromolecules, they're too large to diffuse through the plasma membrane, therefore water moves instead. We still want equilibrium. If the solute molecules can't move, water will move instead. Okay, so active transport. When you think about it, if I'm going to go out and be active, I'm going to need an input of energy. Active transport, when we're looking at movement of solute, is exactly the same. It requires energy. So the movement occurs up the concentration gradient. So in my cell, I've already got some potassium, but I want more. So I'm going to put an input of energy to push more potassium into my cell. It occurs via proteins in the membrane. So those are the guys that are embedded that give it the mosaic look. It occurs faster than diffusion. Why? Well, I'm inputting energy, so I'm kick-starting the process. Okay, when you get a question like this on your exam, don't look at it, think, I hate maths, and keep going. What you need to do is stop, read the question, and walk away with full marks. So, What this question is asking you is what type of transport would be required to move the following. So for the first one, I want to move sodium from seawater, which is 500, to marine alga, which is 100. So I'm going from an area of high concentration to an area of lower concentration. I'm going from higher to lower, therefore it's just going to be simple diffusion. If I wanted to move sodium from marine alga to seawater, so again I've got sodium from marine alga, which was 100, to seawater, so from 100 to 500. So I'm going from an area of low concentration to an area of higher concentration. Therefore, it's going to be active transport. If I want to move calcium from fresh water, so calcium in fresh water is 0.5, and I want to move that to pond alga, which is 10. So I'm going from an area of low concentration to an area of high concentration. So I've already got calcium in my pond alga, but I still want more. Therefore, I'm going to have to input some energy, so it's going to be active transport. And the last one, I want to move chloride from pond alga, which is 100, to fresh water, which is 1.0. So I'm going from an area of higher concentration to one of lower concentration, so that will be a passive process. So questions like this, just stop. Don't worry about the numbers, and if you've got no idea, just read it step by step, and chances are you won't even need to apply any maths, only if you know one number is bigger than another number. That's it. Okay, so with bulk transport, so if I have got something that's really big that I need to get into my cell, so protein synthesis, so a cell makes protein, so the instructions are sent out by the DNA in the nucleus. Proteins are assembled by the ribosomes around the endoplasmic reticulum. They're modified by the Golgi, and then they're ready for export out of the cell. So if I want to export a protein out of the cell, I use the process of exocytosis. EX for exit. So you get these little buds form, what we call vesicles. They bud off from the Golgi, and what they do is they attach to the plasma membrane. So once they've attached to the plasma membrane, they then open up and their contents are released into the extracellular environment, so outside of the cell. Conversely, or from the opposite end, I want to bring something into my cell. So a neighbouring cell's made some proteins for me and I need to bring them in. So my plasma membrane opens up, protein goes in, the vesicle then seals, buds off, and then that protein is brought into my cell. That is by endocytosis, EN for enter. So when we look at bulk transport, so the export of secretory macromolecules, so big molecules, is by exocytosis, and engulfing material, so bringing it into the cell, is endocytosis. This occurs across the plasma membrane in the form of active transport which requires energy. If I think about it, so if I'm going to transport something in bulk, I would need a truck. A truck is going to need a lot more energy to transport than a car. So exocytosis and endocytosis, bulk transport, is going to need an input of energy. These processes are unique to eukaryotic cells. So you see the word eukaryotic, you need to think either a plant or an animal cell. It's more complex, it's got membrane-bound organelles, it's got nucleus, DNA housed in a nucleus. These are the things, so start going through your checklist, so on the exam these things are automatic. So you need to think, how are protein molecules secreted or exported from the cell? Okay, so we're thinking about protein synthesis. I need to know, if I'm going to be exporting proteins from my cell, how do I actually make them? So, ribosomes, you heard about these guys in Year 11. They are structures that lack membranes. They are part protein and part RNA. They are the site of protein synthesis, so they are essential to your survival. In prokaryotic cells, so simple cells such as bacteria, ribosomes float freely in the cytosol, so the liquid component of the cell. In eukaryotic cells, they can be found free in the cytosol, or they can be attached to the walls of the endoplasmic reticulum. One thing to remember, endoplasmic reticulum and Golgi, they look very similar. Your endoplasmic reticulum will always be found near your nucleus because it's involved in protein synthesis, whereas your Golgi will always be found near the outer membranes because it's involved in transport in and out of the cell. One thing that you may notice, you don't need to know about this just yet, but it helps if you can store it somewhere in the memory bank. Your ribosomes have a large subunit and a small subunit. When we go through protein synthesis in more detail, that will become important. So the next organelle that we need to know about is your endoplasmic reticulum. So they are a series of folded membranes and tubules found in the cytoplasm. If it's rough, it's got ribosomes attached and connected to nuclear pores, so the little holes in the nuclear envelope that allow things to enter and leave. Your smooth endoplasmic reticulum produces lipids and steroid hormones. So proteins produced by the ribosomes enter the endoplasmic reticulum tubules and they are transported throughout the cell. One thing to know for your exam, you cannot shorthand the word endoplasmic reticulum unless you write the whole word, then in brackets put your abbreviation. You have to do that for every single page of your exam. So if it was me, just write the whole word. In the long run, it's going to take you less time and effort than to make up codes. Okay, the area within the endoplasmic reticulum membranes is a cisternal space, so the little gaps between the folds. So our Golgi, these look very similar to endoplasmic reticulum, except they never have ribosomes attached. You can call the Golgi either Golgi Body or Golgi apparatus, both are correct. The Golgi is spelt with a capital G. Their job is distribution and shipping for the cell's chemical products. So things that one cell makes, it can then export to another cell. It modifies proteins and fats built in the endoplasmic reticulum and prepares them for export to other cells. The materials that are transported are done so in little vesicles, so little buds that come off, and sugars are added to proteins to make glycoproteins. Okay, so the steps in the protein secretory pathway, this has been on exams in the past. So if you can learn this, and things like this, I try just to learn in a logical way. So you sort of think, what makes sense? So if I want to secrete a protein, first of all, I need to make it. So my polypeptide, poly meaning many, peptide is a type of bond that we have with amino acids. So a polypeptide is produced on the ribosomes. Step 2, my polypeptide enters the endoplasmic reticulum tubule. It is then modified in the tubule and enclosed in a transport vesicle. So I've made my protein on my ribosomes, it's been modified, now it's ready for export. So step 5, it is further modified in the Golgi and then it is ready for export from the cell. So I've got my Golgi, we already know that my vesicles can bud off. So my protein is placed in a transport vesicle and then it is sent over to the outer membrane for release into the extracellular fluid. So here just looking at exocytosis again, so exo to exit. So it's a process of transferring material out of a cell. So a small vesicle fuses with the cell membrane and that enables this to occur. These vesicles may contain enzymes, hormones or antibodies depending on what is actually produced within the cell. It does require energy, so when we are looking at eukaryotic cells, the energy is in the form of ATP or adenosine triphosphate. So endocytosis, EN, to enter, is the opposite of exocytosis. So I'm bringing something into the cell. So for example, another cell has made a protein or a hormone, my cell needs it, so I need to import it. So it usually involves materials that are too large to directly pass through the membrane. So things such as, your large carbohydrates, proteins. So the way that it happens is a small vesicle buds from the cell membrane to enable this to occur. Again, it does require energy. What type of energy? ATP, adenosine triphosphate. If I am ingesting something into the cell, it's also known as phagocytosis. That's a word that if you can start to incorporate into things like your chapter questions, then when you get to your exam, your language will be more complex. Now, the one thing with biology is our cell structures are actually designed to meet the needs of our bodies. So we don't waste energy doing things that we don't have to do. So the processes within a cell are due to molecules interacting with each other. So to interact, they need to actually come into contact. Molecules need to move into and around the cell at a certain rate to reach sites of specific activity. It's a mouthful. Molecules need to be in adequate concentrations if chemical reactions are going to occur at the right rate. So if I need to make a polypeptide chain, I need to actually have the sufficient materials to build it. So the molecules need to come into contact with each other, or else that is not going to happen. So cell structure needs to facilitate or assist the movement of molecules and maintain them in adequate concentrations so reactions can occur to maintain cell functioning. This is where osmosis and diffusion, bringing things into and out of the cell, comes into it. So movement across the plasma membrane, cells need to maximise their surface area to ensure the rapid movement of molecules across the cell surface. If you think about it, so at some point you've probably done food tech and you've had to melt butter. If I put a block of butter into a saucepan, it's going to take a much greater amount of time to melt that butter than if I cut it up into small pieces. Every time you cut butter into smaller pieces, you're exposing more surface area. So with my plasma membrane, I need to have sufficient surface area to enable the import and export of required molecules. So when we think about movement within a cell, we can compare prokaryotic and eukaryotic cells. So cells need to obtain nutrients that they require for their survival and also to get rid of wastes. The higher the surface area is compared to the volume, the easier cells find it to absorb or lose substances. So if I compared a mouse to an elephant, even though an elephant is a much larger animal, it has much less surface area to volume compared to the mouse. So it's got much less outside compared to the bulk that it is covering. So to maximise your surface area to volume ratio, many structures are folded and or flat. So for example, when you look at your mitochondria, they've got all those inner folds that when we look at, say, the respiration, we've got a heap more surface area for things to occur on. So cells can't be too large. If they are too big, they would not be able to survive. The membrane wouldn't be able to support the actual cell because it required too much energy. So it would be too costly in terms of the time taken to transport substances within the cell. So it'd be like you being dropped off to school in the western suburbs every day. It wouldn't be feasible because it would take too much time out of your day and too much energy. That's why most of you are going to school locally. So to overcome this, larger organisms are multicellular. So they're made up of many smaller units. You look at yourself, you're made up of billions of different cells.